Good afternoon, everyone. My name is Ibrahim Alsaggaf. I'm a PhD student at Birkbeck, University of London. Our team CSBBK ranked in third place in this DREAM challenge. On behalf of the team, I would like to thank the challenge organizers for giving us this opportunity. Let's start. Okay, let me flip this. All right. Okay, brilliant. Let's start by talking about some key aspects of this challenge. Of course, the task and the data has been explained in the previous talks, but I will cover this part from a different perspective. As you know, both the training data and the leaderboard data were generated from the same studies. Therefore, they should share the same batch effects. On the other hand, the testing data was generated at a single center, I would say a homogeneous population. Another key aspect is understanding the data characteristics. The raw beta values matrix is high dimensional, 50K and dense, so it isn't sparse, and its values lie in the range between 0 and 1. And this part of information is crucial, as you will see in the coming slides. For the gestational age, which is our target variable, as presented before, it has a long tail distribution, and its values lie in the range between 5 and 44, which is again quite a large range. Due to time constraints, as we joined the competition halfway through, we sought a simple yet effective approach. So our assumptions was this approach should not rely on domain knowledge, should not require any expensive trial and error processes, and such as heavily pre-processing tasks or maybe feature selection. And basically, I would say it's a simple data-driven approach where most of the information is maintained. Additionally, it should handle high dimensional and imbalanced data. Okay. So here I would like to introduce an emerging type of representation learning, which is called contrastive learning. So this type of representation learning simply works as projecting samples that have similar features to a target hypersphere at close locations, whereas other samples that have different similarities or different features should be projected at far locations on this hypersphere. This learning paradigm showed its success in single-cell RNA-seq analysis, and several works have used contrastive learning in batch effects removal. Recently, our works showed that contrastive learning successfully improved the accuracy of cell type prediction tasks, outperforming other well-known state-of-the-art prediction methods. So now in the following slides, we focus on addressing those key aspects. We applied a standard pre-processing step. Thanks for the challenge organizers. We utilized their provided R script to filter out those probes that fall near SNPs aligned to multiple locations or target locations on X and Y chromosomes. We also used the same script to filter out probes that are not reliably detected in all samples. We set the p-value cutoff to 0.01 following their script. We used the filtered raw beta values to train our model without any normalization or feature selection. For the gestational age, as we have seen, the range of values is quite large, so we simplified the task by scaling the values to lie between 0 and 1. And this is we used a conventional min-max scaler. So our approach involves two staging of training. The first stage, we adopted self-supervised contrastive learning to learn some representations Of course, those representations will be invariant to the imbalance ratio in the gestational age, because those continuous numbers were not involved in this stage of training. And we hope that we should learn some new space where those samples that have similar features lie close at each other. So a sort of clustering. And here we assumed that those samples that bear similar features also bear similar gestational ages. We used a simple architecture here, nothing complicated, just a multi-layer perceptron encoder and a projection head, and also MLP projection head. For the augmentation method, and here the use of augmentation in the context of contrastive learning is to create new different views of a given original sample. So we use an augmentation method that utilizes two different Gaussian distributions to create two different views of each original sample, where those views are used for the contrastive task. And here I would like to sincerely apologize for not further explaining the details of the augmentation method. Our paper is under review, and the journal has strict requirements prohibiting us from sharing or discussing any of this in the press or elsewhere. So for the second stage of training, we replace the projection head, just dump it, and we use a regression head instead to fit the task in hand. And similar architecture, just one fully connected layer with a single output layer to opt a continuous number. Here we treated our encoder that was trained in a self-supervised fashion as a pre-trained model, and we fine-tune it along with the regression head on the scaled target variable. Here for the loss functions, I know this slide is ugly, but I will go through it quickly. So for the loss function, we aim to minimize the conventional self-supervised contrastive loss for the first stage. And here, if you see in the numerator, Zi and Zj represents the two different views of the original sample. And for the second stage, we use the well-known root mean square error function, but we did a small alteration by adding a small number just for numerical stability during the backward pass. During the 30 epochs of training, we save the model every epoch and then use those saved models to predict the first 100 training samples. Those samples were just picked randomly. After that, we inversed the prediction back to their original range. So from 0 to 1 back to 5 till 44, the original range, and then measured root mean square error against the ground truth. Then we selected the model that obtained the best score, which is the model at the 28th epoch. Of course, we utilized the leaderboard data as a held-out validation set. Our model ranked in 3rd place in the final round and ranked in 4th place in the leaderboard round. This demonstrates that our model has good generalization and showed cross-study robustness. Yet, it would really interesting to merge domain knowledge with our approach and see what new insights could we discover. And I apologize for not sharing any results, again, due to time constraints, but I would love to share that to show the new representation space and how those samples were grouped or so-called clustered. Finally, we would like to thank the DREAM Challenge organizers for covering the conference registration fees and for hosting this event. And we would like to thank the School of Computing and Mathematical Sciences at Birkbeck University of London, for their GTA program. Thanks for your attention, and I'm happy to answer any questions.
Ibrahim, can you hear us?
Yes, I can now.
Okay, so thank you for your talk. I have a quick question. So we typically try to look in the end to see which features eventually coming from what genes in this case were involved or had more importance in the model. Do you have a way to assess that, at least based on your first encoder?
Yes, there is a way that involves gradient to interpret which features has the most, I should say, influence on the decision. This is possible because the network is actually very simple and nothing complicated about it. Just feed forward network, and having this piece of information is applicable. Sorry, I can't hear you.
So we don't have other questions from the audience. The other thing I was wondering, so were you able to run the model with the 450 and 850? And if you're not able to run with the 850, would it be possible to maybe run a separate one just with the features that are only on the 850 but not on the 450? So in other words, a way you can help to answer this question, is this higher coverage useful or not?
Okay, for the 850, sadly, to unprecedented circumstances, we couldn't use our department computing resources. That's why we just stick with the 450. Actually, I only use my laptop. And for the 850, the plan was to just mask those should say null values with zeros, because I know this approach is powerful against sparse data. So having this sparsity wouldn't be an issue, and the model should look its way through and find a reasonable solution.